Okay, so I'll give again a very basic introduction and then introduce the speaker. I also wanted to catch us up on where we're at with this experiment in CASP 16. So this is our special interest group on ensembles of conformations. And the concept is that CASP has been very successful in driving the field of protein structure modeling, particularly for relatively small proteins, or anyway, not so much small proteins as static proteins. There still remain challenges with complexes, and particularly with modeling the multiple conformational states of proteins. So we've put together this special interest group to explore this challenge. So I'm going to talk a few minutes about an introduction and the progress so far in this experiment. And then Giulia Palermo has kindly agreed to talk about work she's doing with multiple conformational states, particularly with respect to the CRISPR-Cas9 system. So the special interest group has this vision statement, recognizing that biomolecules don't have static structures, but rather adopt a distribution of conformational states. The CASP SIG aims to evaluate if it's possible to critically assess the accuracy of such predictions and whether it's even possible. One of the big challenges is getting populations of states, particularly if you want to validate against experimental data. So we consider various developments in the field, both computational methods and experimental methods. And hopefully over time, we can define protocols for assessing the accuracy of conformational ensembles. And so what we're talking about is from a simple example of a two-state system. Adenylate kinase is kind of the classic example of flipping between open and closed states, to kind of the other extreme, intrinsically disordered proteins or intrinsically disordered regions of proteins, which may adopt many, many different conformations. And this kind of it illustrates the problem, but also the challenge, because you can always fit data better by adding more degrees of freedom. And so this is generally one of the challenges in this field. So we have a steering committee who I work with to consider who to invite to present, and they're listed here. Shoshone is on the call, and I think John Molt is also on the call, and Andre, and maybe some others by now. So John and Christoph and Andre, they give us advice on how to connect this with a real CASP activity. So we do have now in CASP 16 an ongoing experiment on conformational ensembles. And if you go to the CASP site and you click on the link here, ensemble, which comes up right when you come on the site, you get this page. And this lists the various targets. And they're also annotated as ensemble. So these target 3 and 4, it's actually the same protein sequence, but we have two different structures that have been determined experimentally. And so the challenge to the predictors is to model the two states in this case. And most of them are presented that way as two states. Our first ones are actually two states, but we only have one target name because we just decided to do this partway through. So it's target 1, target 2, then 3 and 4 are target 3, 5 and 6 are target 4 of ensembles. So there's about, I guess, 8 targets so far. And there'll be a few more will be released during July. So we'll have about twice as many of these. These are determined by X-ray crystallography or cryo-EM. Some of them are complexes with RNA. Two of them are RNA, in fact, or nucleic acid. And like I said, there'll be about a half dozen more or 8 more during the summer. So this is going well. We have about 70 participants, I think, on each of these target pairs. Most cases, we don't have the experimental structures in hand yet, so we're not quite ready to do assessment. We also have another kind of experiment going on, and it's one that was proposed by Terry Ois, who's on the call, and Bruce Donald and their team. And this is an experiment really focused on the question of intrinsically disordered regions, and specifically intrinsically disordered regions that are between domains, interdomain regions. And so this is the protein A of staph nuclease. And protein A has 5 domains, and these domains are highly homologous and very similar structures to one another. And the domains A, B, C, D, and E are connected by short linkers. And so the challenge here is to model the interdomain structural variability. And so it's really a modeling task on the interdomain linker. So it's our first foray into really trying to predict conformational distributions of intrinsically disordered regions. And what Terry and Bruce have done is to develop a system where you can take a pair or more of these domains and determine the relative orientations of the domains with respect to one another. And that's done, and I think it's shown better here. You have 2 domains, and there's some connected linker, and you can put an orienting metal in one of the domains, and that orienting metal, covalently attached and very low mobility, orients one and causes the creation of an NMR parameter called the residual dipolar coupling that gives information about the orientation of this domain relative to the applied field. And then you look at the RDCs in the second domain, and if the second domain is strictly oriented by the first domain, it will have the full range of RDCs that you'd expect. But if there's motion of the second domain relative to the first one, it'll attenuate those RDCs. And by looking at that distribution, you can model the relative kind of model independent assessment of the relative orientations of the 2 domains. So that's actually the CASP challenge we're going to be putting out in July. And there's a paper describing the work they did to develop a method for representing relative domain orientations and how to extract that information from the RDC data. One thing that's very critical here, and you should listen if you want to participate in this particular CASP experiment, to Terry and Bruce's video, CASP SIG video of March, which is online in YouTube, about some details. And I think there was even a follow-up one where more details were provided. The instructions will explain these have to be treated as rigid bodies and modeled exactly the same as they are in the experimental structure, so that the orientation axes can be properly defined. And so the models will be variable based on the interdomain linkers. And that's going to be explained in detail in the instructions that are provided with this target. So this is a different kind of target. Terry is also generating, together with Susan Tsutakawa at Berkeley, SACS data on the same system. And the SACS data is not as, maybe not as detailed as what you get from these NMR studies, but it also gives information about relative domain orientations. So we can also back calculate SACS data from the ensembles that predictors provide and assess the fit. And I should also emphasize that a challenge here is not only what are the conformations, but what's their populations? Because if you don't have the right populations, you won't be able to back calculate the data, or the data won't be back calculated properly. Terry, I know you're driving, and I don't want to do you want to just interject it all there, or we just should go on now?
No, you did a great job, Guy. Thanks for summarizing. And yeah, you said everything that I think I would say. Thanks.
And we actually have a second target like this. It was actually designed to do the same NMR experiment, but we won't have that NMR data before August. but the end of August. But we will have the SACS data, I believe. The sample's been sent to Susan. So this is a second two-domain system with a flexible linker. This is actually the integrase for murine leukemia virus, and it binds to a host protein to allow the virus to integrate its genome. And this interaction is critical to that, and the interaction creates a linker. And so this is a second of this kind of target that we will release subsequent to the protein A, but during the summer, we'll release this one also. So these are just the models that came out of the NMR structure calculations. They're not anything real about relative domain orientations. We want to do some sort of molecular dynamics or other methods of modeling these distributions, and then we'll compare them against experimental data. So I just say that comments are welcome. The PowerPoints and the video will be made available. We have a LinkedIn group to share information on this general topic of multiple conformational states of proteins. And if you email to this email address with the subject CASP SIG, we can put you on the mailing list. We've been making the videos available, and there is a YouTube channel of all of the ones we've done so far. There was actually an earlier YouTube channel that mixed with some other stuff from my lab, and then I broke off the separate one. So if you don't see the latest one, you might not be on the latest YouTube channel. But anyway, here you can hear about, and particularly, this is the Ose and Donald SIG presentation. And I just mentioned that this is one of multiple special interest groups that CASP has set up. There is a CASP special interest group specifically on AI methods in molecular modeling and in biomolecular modeling that Arnie Olafsson organizes. And this is on the first Wednesdays of, that's here. This is on the second Wednesday of each month. But they're taking a break for July and August. So I think the next one is in September. And there's also a special interest group on modeling RNA that's organized by Rachel Kretsch. And here are links to the targets and to the meetings and the YouTube videos. And we do have a CAST meeting scheduled November 29th to December 3rd, but we haven't settled the location, whether it will be in, it's most likely going to be in the Caribbean, but the location hasn't been settled yet. So with that background and introduction and update, it's a pleasure to introduce Giulia Palermo from the Department of Bioengineering and Chemistry at UC Riverside. So Giulia has been doing really beautiful work on using molecular dynamics, particularly to model multiple conformational states of proteins and enzymes, and connecting this with experimental data, particularly cryo-EM, NMR, and crystallography data, particularly some very nice work connecting with NMR data. I think it's very appropriate now to have a talk. Most of the talks so far have focused on machine learning methods for modeling protein structures, but molecular dynamics continues to be a very important approach, and there's synergies between them. And Giulia has evolved to be one of the leaders in this field. So I'm very pleased that we have this chance to have a presentation. So Giulia, do you want to pick it up from there?
Thank you very much for the kind presentation. I'm going to start sharing my screen. One moment. So first of all, I would like to thank you very much for the kind invitation and for giving me this opportunity to present here some of the research that we are doing in my lab. Today, I will talk about how we are using...
Want to swap the displays so that we can see the full screen. Here we are seeing the your next slides.
Okay.
Yeah, great.
Thank you.
Thank
Okay.
you, Emily. That was very helpful.
Thank you very much. So I will tell you how we are using computational methods to characterize function of genome editing systems, and also how we plan to engineer them. This slide is taken from a recent paper by Wang and Doudna, and it shows the future directions of the gene editing field. So this is where we could be in the near future and in the more distant future in terms of manipulating nucleic acids. What we can do, we can impact agriculture with drought-resistant crops, impact biomedicine, impact material science. Of course, as you see from this perspective, at the core of this, there is design and engineering of improved gene editing systems in which computations can have a very big role. And this is why we call this field computational genome editing. So far, computational drug discovery has been very successful. From 40 or 50 years, computational drug discovery has been tremendously successful with the invention of new drugs and therapeutic molecules. However, to understand and engineer systems that manipulate nucleic acids is much more complicated with computations, because they have very long time scales. They are characterized by very long time scales. They are very large. And the mechanisms that we want to target are not intuitive. So this is why my lab wants to embrace in this journey that is called computational gene editing in the long term, based on the first yearly publications and the publications of my lab. And the publications of my lab, here is where we are in 2024, where we characterized CRISPR-Cas9 systems, engineered the gene editing systems. This is where we want to go in terms of data-driven modeling, multi-scale models, to ultimately employ computations for personalized medicine. So as Gaetano has introduced, the role of deep learning and AI will be absolutely central to design the genome editing systems of the future. However, a physics-based approach, which is what we are more expert in, will help understanding the function and will feed again design. At the foundations of computational gene editing, there are the studies that we have done during these years. And this is an outline of my talk in which I will give you a little bit of background and significance of CRISPR-Cas9. Then I will dig into the biophysics of gene editing, so how nucleic acids bind and achieve selectivity. So the gene editing revolution started in 2012 when Doudna Charpentier discovered that this protein Cas9 can be RNA programmable, and it basically works by first binding a guide RNA and then using this sequence to recognize and cleave any desired DNA, which then gets site-specifically cleaved. Thanks to the recognition of a short sequence in the DNA called PAM, the system can basically target any DNA you want. And for this reason, it has been harnessed for gene editing with modifications in animals, in medicine, in biotechnology, even in material science. However, when we started our research, there were a series of open questions characterizing the biophysics of Cas9. Cas9 is a protein that changes conformation very rapidly, similarly to an aerial cell artist that first grabs the DNA, changes conformation to bind the DNA and match it to the guide RNA and perform cleavages. However, through this complex conformational activation, several questions have been unclear, such as the allosteric effects characterizing the protein activation, the binding to off-target sequences, the catalysis, how does it cleave, and so on. So we started our research by first characterizing the conformational activation. And this has been a complex study in which we studied how the system changes conformation from APO to DNA, to RNA and DNA bound. But I would like to focus on the conformational activation of the catalytic H&H domain, which was found to assume multiple inactive conformations through structural biology. In this case, we used a Gaussian accelerated MD method and Anton 2 simulations to characterize how H&H changes conformation based on the 3 inactive structures that have been interpolated, while also characterizing what we called a putative, a bona fide model of the active state, where H&H docks at the cleavage site. You can see this was identified with accelerated MD and with Anton 2 simulations. And a couple of years later, this first study, when the structure of Cas9 in an activated form appeared, the agreement between the predicted model and the cryo-EM map was quite remarkable. So the reason why I have introduced the dynamics of H&H is because the dynamics of H&H, according to these 2 seminal papers appeared in Nature, is extremely important for the entire activation of Cas9. So what happens, according to these papers, H&H would change conformation, and through this conformational activation would activate in an allosteric way the other catalytic domain, RAV-C. However, the mechanism by which this happens was unknown. It was also suggested that binding of the recognition sequence would activate this allosteric crosstalk. But again, it was unclear how this would happen. So we decided to perform molecular simulations of Cas9 based on 2 structures bound to PAM and in the absence of this recognition sequence to basically trace the allosteric communication. And to do so, we employed network models derived from graph theory. In these models, you can describe your system as a graph of nodes and edges, where nodes are amino acids and nucleobases. The bond between these nodes is basically weighted on correlations. So the stronger the correlations between 2 nodes, the closer these residues. And these are the same models used in artificial intelligence models applied in Facebook and Twitter, where you have multiple friends and connected by edges. Some of these friends display more connections, and the communication thereby passes more efficiently through these friends. And basically, these are the friends to whom you don't want to tell your secrets. The same models can be applied to proteins to identify the residues that govern the conformational dynamics, that hold the majority of correlations with other residues. By applying algorithms such as Digistra, we can also describe what are the shortest path of information from one specific site to the other. For example, H&H and RAV-C, the 2 catalytic domains. This model basically maximizes calculates the paths that maximizes the correlations from one site to the other, and thereby minimizes the route among them. And these are the results from our yearly papers. What we see is that when we introduce the recognition sequence in the simulation, H&H and RAV-C get correlated with each other. In the absence of PAM, this correlation is lost. This is already a sign of an allosteric activation. This is a typical sign of allosteric activation that in literature is reported, for instance, for GPCR, when an allosteric effector binds to a protein, shifts the conformational ensemble with a change in correlated motions, which is clearly observable in this case when PAM binds. We then structured our network. This is the allosteric network that we computed, showing a strong flow of information from the recognition sequence, the recognition lobe where the DNA binds, to the catalytic sites. And we also identified the key residues that transfer the information, the key residues that hold the dynamic exchange between HNH and RAFC. What was interesting is that alanine mutations of these key network nodes improved the specificity, was shown to improve the specificity in one paper published before us, and in 3 papers, in particular, Schmidt-Borg was inspired by this observation that L1 and L2 transfer the dynamics, and specifically targeted the mutation of these loops. Also, Bravo, in a very recent paper, finds that alterations of the L2 loop can improve the specificity of Cas9. So this opens an important hypothesis, the hypothesis that allostery has something to do with specificity, or that CRISPR-Cas9 improves the specificity against off-target effect by leveraging the communication between the recognition site and the nuclease site. So that's why we decided to start studying this specificity enhancement of Cas9, starting from 3 mutations in HNH that improve the specificity. We focused on these 3 lysine to alanine mutations that are located in HNH and are adjacent to a pathway that we identified to maximize the communication between the recognition site and RAFC. So using solution NMR, in collaboration with George Lisi at Brown University, we observed that there is a pathway of slow residues, shown here in red, that connects the recognition site, so the site where the DNA binds, to the sites where the DNA cleaves. We then structured HNH through community network analysis in communities of highly correlated residues. And some of these communities, which we called allosteric, are the communities that host these slow residues. By computing then the difference in edge between us, so what can be defined as the traffic between communities, we can see that when we introduce the mutation, there is a loss in communication between allosteric communities. To verify this through this observation, through solution NMR, we combined our community network analysis with CPMG relaxation dispersion. So what you can see here in these graphs is an NMR-derived communication network, where the communities are derived from computations, but the bonds, so the communication between these communities, comes from the experimental dynamic exchange, so from CPMG relaxation dispersion. And you can see that when you introduce the mutations, clearly the communication between the allosteric communities is reduced, as it was observed through graph theory, through the analysis of the dynamics from graph theory. So these could tell us that allostery can be a source of specificity enhancement, which prompted us to study how do mutations that are located in REC, in this domain, that is distal with respect to HNH, the catalytic domain, alter the dynamics of HNH and improve specificity. It was shown that these three systems, hyperaccurate, high fidelity, evolved Cas9, which hold mutation in REC3, can alter the dynamics of HNH and improve the specificity of Cas9. This is taken from Chen et al., showing through single molecule experiments that in the enhanced specificity Cas9, HNH alters the dynamics, alters its dynamics in the presence of off-target sequences with respect to the system on target. So we wondered why, we wondered what is the communication, how mutations in REC alter or communicate with HNH. And we applied our graph theory approach on in this system, in the wild type, and in our enhancing specificity mutants. What we observe is that mutations in REC3 improve the communication from a very interesting site, this helix, alpha 37. Alpha 37 is a helix that was shown in our previous, in one of our previous papers to recognize mismatches within the hybrid. So these mutants reduce the communication between this helix that recognizes mismatches and the HNH catalytic core, providing an explanation on how these variants function. What is actually very interesting is that these variants hold mutation in REC3 at different sites with respect to this alpha helix. And if you compute the communication from these different sites to the HNH catalytic core, you basically don't see a difference with respect to the wild type. But what happens is that the communication between the helix that recognizes the mismatches and HNH is remarkably altered. The next part of my presentation will focus on the biochemistry of gene editing. So how does Cas9 cleave DNA? So Cas9 holds two catalytic domains, HNH and REF-C, which cleave DNA through the aid of metal ions. If we want to study how enzymes perform a chemical reaction, we should go beyond classical molecular dynamics because we need to break and form bonds. So we need to explicitly describe the electronic degrees of freedom. So we need to go to quantum mechanics. And of course, we need to do this along molecular dynamics. We need to sample the states from the reactant to the products using molecular dynamics, using either a Born-Oppenheimer approach, where at each step a Schrödinger equation is solved, the forces are computed, and then the dynamics move forward, or by applying some tricks that enable us to speed up the dynamics by treating the electronic degrees of freedom as semi-classical particles, which enables us to basically not always solve Schrödinger equation, and basically we can go faster with the dynamics. So we employed both methods, though in this case, this approach, the Carparello approach, worked really well. So we had excellent results. We did not need to go to Born-Oppenheimer. Okay, so let's talk about the RAF-C active site. When we started these investigations, we had no idea on how the magnesium ions would bind. We started our investigation based on a manganese-bound structure without the substrate. So through our approach, we identified that an arginine residue preferably likes to bind the schissyl phosphate, likely acting as a stabilizer of the active site. And we also found that this histidine acts as an activator of the nucleophile and basically activates the nucleophile for phosphodiester bond cleavage. So these observations were in line with the available kinetic experiments. The profile was in line, but what is more interesting is that David Taylor obtained very recently a structure of HNH with the ions structured in the active site right after phosphodiester bond cleavage, showing that the active site that was predicted was remarkably good. This is instead the study of the catalytic HNH domain. In this case, we used metadynamics simulations to study how HNH cleaves DNA. In particular here, again, the histidine is the activator of the water nucleophile, which leads to the transition state and products. This is a 2D free energy profile going from the reactants to the products obtained through metadynamics simulations. But what is more interesting, I think, of this paper, rather than the chemical step, description of the chemical step itself, is that prior to our study, a couple of configuration of the active site were identified. One configuration showed two aspartate residues coordinating magnesium, but another configuration, which was supported by this cryo-EM structure, showed that an asparagine residues would take the place of the aspartate through a conformational change of this back loop to possibly coordinate magnesium. So people believed into the pseudoactive state in which the two aspartate coordinate magnesium, because the majority of the crystal structures and of the cryo-EM structure supported this model based on the homology with another nuclease. However, this cryo-EM structure obtained with magnesium was clearly showing electronic density, and it was unclear, basically, why prior studies supported these the other active site. So what we did was to perform umbrella sampling, moving from the pseudoactive to active with magnesium and without magnesium. And what we see is that without magnesium, the pseudoactive configuration is favored, but while with magnesium, the active configuration is favored. So this showed that magnesium holds a structural role, leading the system to assume an active conformation. And also clarified why the previous states of the previous structures, which were obtained in the absence of magnesium in the experiment, fell into the characterization of the pseudoactive conformation. This is a movie of the catalytic mechanism of target DNA cleavage, showing you how the catalysis works. So here you can see the system. The system is very large. It's approximately, this model was approximately 400,000 atoms. I would like to mention that now we are digging into the active site, how it is organized. So this is the largest system studied with QM-MM simulations. Simulations of the spliceosome, which is larger, have been performed, but on smaller models. So this is here you see the transition state. We reached the transition state, bonds partially formed and cleaved. Now the bond cleaves, and the O3 prime gets protonated by the lysine, which was also an interesting mechanism not observed before in other studies, and was corroborated by biochemical experiments, DNA cleavage experiments, and was also corroborated through additional quantum simulations at higher level of theory to basically back up the limitations of the DFT functional, of the DFT functional we had to use to propagate the dynamics. Okay, now let's go to the specificity of gene editing systems, which is about the onset of off-target effects. So on-target effects are the unselective cleavage of DNA strands that do not fully match the RNA guide, bearing base pair mismatches. This work was inspired by a paper by Chen and Agnes. In this paper, through single molecule experiments, they have shown that in the presence of 4 base pair mismatches at the end of the hybrid, HNH assumes an inactive conformation, while when we introduce 3-2-1 mismatches, HNH assumes an active conformation similar to the on-target system. So we wondered why. So what we did is to perform molecular dynamics simulations on the conformational checkpoint, so a state of Cas9 in which HNH assumes an inactive conformation to mimic the experimental setup. We also mimicked the experimental setup by introducing the same type of mismatches that they have introduced into the experiment. So we saw that in the presence of 4 base pair mismatches, the hybrid remarkably opens, establishing contacts with the L2 loop. So establishing contacts with the L2 loop, locking HNH into an inactive conformation, remarkably reducing the conformational dynamics. And this is a movie of the locking interactions on how HNH gets locked by the target strand, including mismatches. It's also very interesting to note that the very recent super phi Cas9, so the variant of Cas9 that includes mutation in this loop, basically minimizes the mutations with this, alters, sorry, the mutations with the hybrid. So the super fidelity Cas9 improves the specificity by altering this loop that we have shown to basically be locked by the mismatches in Cas9. So these, based on these observations, Akash Shaha worked with Martin Jinek on the characterization of how mismatches would look like along the hybrid. Martin obtained structures of Cas9 with adenine-adenine mismatches at specific locations, and Akash, based on the agreement we had for the mismatches at the end of the hybrid, was able to map how the mismatches would look like along the hybrid, complementing the structural data. I would like now to say a couple of words about computational engineering of Cas9, how we are engineering these systems for improved gene editing and detection. So these studies have been focusing on systems including Cas12a. Cas12a is an additional gene editing system that was found to have a very interesting property. So through a very rapid conformational change of the protein, this protein is able, after recognizing DNA and performing double-stranded DNA cleavage, is able to remain activated and cleave any single-stranded DNA with a reporter. So for this reason, this system paved the way for nucleic acid detection, a property that is very important, for example, for the detection of infectious agents, and can be very important, for example, for detecting various infectious diseases. Akash started his PhD in my lab by first analyzing the conformational dynamics of CASP12A. So he published a first paper in which he learned all the basic tools, molecular dynamics, principal component analysis, to characterize basically dynamic switch induced by DNA in the system. Then Akash also worked with a group to obtain a model of transcleavage, the property by which Cas12a remains activated to perform this cleavage of any incoming single-stranded DNA. This process is called transcleavage. But what Akash was most interested in is to understand how can a single catalytic domain, as opposed at odds with Cas9, a single catalytic domain, cleave both the non-target strand that lies within the active site and the target strand that was observed in structured spatially distant at 30 angstrom away. Some sort of conformational change, conformational activation should have happened for the target strand to traverse this distance and reach the active site. So what happens here is that Akash first used Anton 2 to characterize the long time scale dynamics of the system, and he found that an element in RAPC, an alpha helical lead that was supposed to close the RAPC active site, close and open the RAPC active site, was engaging in strong interactions with the target strand, leading the target strand to approach the catalytic site. Akash also performed umbrella sampling simulations to to basically drive the target strand toward the active site into the wild type Cas12a, which is here in blue, and by the profile is here in blue. You can see that it freely flows here toward the active site, and by mutating key residues in the lead that interact with the target strand, observing that by mutating these residues, the traversal becomes more difficult, especially in the first part when the lead has to bind to the target strand. Akash also performed alchemical free energy perturbations with soft core potentials to basically measure what would be the contribution of these residues into the free energy for target strand binding. And he found that a couple of residues are particularly important. Martin Jinek, through DNA cleavage experiments of the target strand, observed that mutations of these two residues in alanine remarkably reduces the cleavage of the non-target strand, basically counter-proving that this mechanism, so the alpha helical lead grabbing the target strand, bringing in the active site, is a functional mechanism for the cleavage of the target strand. As I said, Akash also worked in collaboration with an Italian group to provide a model for transcleavage, and what they found is that binding of a stem loop is favored with respect to a single-stranded DNA, which is in agreement with the DNA cleavage, with cleavage experiments, showing that Cas12a is faster, performs transcleavage in a faster way in the presence of a stem loop. So a stem loop is a better substrate for transcleavage. Akash also, my group also worked on a novel variant for called CASP SIG. And the leading lab member here was Dr. Shubik Sinha, a postdoc in my lab, a very talented postdoc in my lab. So CASP SIG is another very interesting gene editing system. The reason why CASP SIG is so interesting because CASP SIG targets RNA and can be used for RNA imaging and RNA detection. What was interesting of this system is that based on biochemical experiments, an allosteric mechanism was proposed. According to these experiments, the binding of a target RNA would activate the catalytic function of this distally located active site composed of the HEPN1 and HEPN2 domains. And the mechanism by which this occurs was clearly elusive, because how can something binding here activate cleavage of any incoming nucleic acid on the other side? So initially our study was meant to characterize this allosteric communication. We wanted to understand why and how. But while we were investigating this mechanism by computing the shortest route communicating the target RNA and the active site, and also developing this measure called signal-to-noise ratio of communication efficiency, calculating how the signal stands out from the noise, we identified critical residues in the communication, which we thought would have been important to transfer the information of RNA binding to the active site. We also corroborated our investigations through a machine learning approach, and Mitchell O'Connell from the University of Rochester performed experiments, including these mutations, and observing that these mutations make CASP-13A a more specific variant against single nucleotide mismatches. This is the premise of our study. In particular, here we saw through computational investigations that these residues increased interaction with a critical adenine from the CRISPR RNA, and that's the reason why we thought they were so important. By applying our signal-to-noise ratio, we see that the signal is remarkably dampened when these mutations are introduced in the presence of an anti-tag RNA. And here you can see the experiment that basically has set up the premise of our study. So the fact that our variants can select a target RNA over an anti-tag RNA. As I said, this was the premise because then Mitch introduced mismatches within the system at specific positions, observing that the system can selectively perform or not cleavages according to the location of mismatches. So coming back to the future, I would say that I hope I have convinced you that our approaches can be useful for both designing the gene editing systems of the future and to understand their function to feed up again design. And with this, I would like to thank all of you for your attention.
Well,
and I would like to thank my lab, who has been instrumental in performing this research. In particular, current lab members, Pablo Arantes, Mokhtar Hassan, Shubik, then Akash, who is a graduate student that is now moving to Columbia University for his postdoc to work with Professor Barry Honig, and the other lab members, Aman, Chinmay, and Kazi, who was visiting our lab. Of course, Lukasz Nierzwicki, who went back in Poland, and all the others. Thank you very much.
Thank you so much, Giulia. That was really great. It really a great demonstration of why multiple conformational states are fundamental in biology and also in biotechnology. So it was really beautiful. I have some specific questions, but I want to give other people a chance to ask questions. So, Shoshona, I see you turning on your screen. People can raise their hands or put it in the chat or just speak out. Go ahead, Shoshona.
It's wonderful. Thank you very much for this incredible talk. You know, you have made so much progress since the early work that I was privy of in 2017 at the CCAM workshop. So congratulations, that was great.
Thank you very much.
The system is fantastically complicated. So, you know, what you have done is awesome. So I have a question. I have one question that I'll ask. What is the correlation between the, I would say, the range of mismatches with the conformational adjustments or conformational sampling of the different domains, and which one are, how are they correlated? Because this is the secret of specificity, right?
Yes, so this is a very good question that actually can open a lot of new research in the lab. Thank you very much for this question. So when we studied, I've shown only a couple of slides about the study on DNA mismatches. So basically here, what we did was to include mismatches at the end of the hybrid and compare to a on-target system. So what happens here, I want to say this thing that I think is very important. So everybody of us know that the force fields for RNA and DNA usually lead to fraying of the DNA at the end of the duplex. So this typical behavior did not happen in the system with a non-target DNA. And the reason why this is not happening is because the protein around keeps it. Basically, it interacts so well with the hybrid that keeps it in a very firm way, especially this helix, alpha 37. Helix interacts with the hybrid and keeps it very firm. As soon as we introduce mismatches, this helix starts entering into the hybrid and opens the hybrid. So the hybrid can interact with HNH and alters the dynamics of HNH. So in this way, the REC3 domain, which is distally located with respect to HNH, alters the dynamics of HNH. So it alters the correlated motions between REC and HNH change. There is a shift in coupled motions. What is very important to say that the mechanism through which this happens is through this helix. So this helix enters into the hybrid interacts with HNH, so the dynamics of HNH is altered by REC through the hybrid that acts as a transducer of the motions. I don't know if it was clear or if this answered your questions.
Anyway, it seems that you have an explanation, so that's interesting. But you know, you would I would say minimize. So how would you go?
I'm sorry, I did not hear the question because maybe the network went down.
You know, now that you have some kind of explanation of these mismatches, you would like to minimize them, right? At some point.
Yes, exactly. And so as this helix enters the hybrid, what it does, it alters the dynamics of HNH on the other side. Let me one moment reach the slide. Okay. So REC3 is here. The alpha helix 37 enters into the hybrid. This part of the DNA binds to HNH and locks the dynamics of HNH. What David Taylor has done in a paper published in 2022, he mutated this helix, altering the interaction with the mismatched DNA, and this improved the specificity of Cas9.
Okay, wonderful. Okay, thank you. It's very interesting. Okay, thanks.
Thank you.
Yeah, we can take another question.
I have one really quickly. I'm curious how you defined the signal to noise. The signal versus the noise? Is it just like the deviation from the Euclidean distance between the nodes, or how did you do that?
Yes, so to answer this question, I would like to open briefly another PowerPoint presentation to show you how the signal to noise was measured. One moment, please.
Your artwork is wonderful behind you.
Okay. Okay, so this is overall how we basically measure a loss rate through graph theory, which I have already basically described before. So rather than cross correlation, we use mutual information, which provides a better representation. But the way how the signal to noise was defined is this. Okay, so first of all, imagine you have your system with your nucleic acids that have to transfer information to the catalytic site. So the limitations of shortest path analysis is that they define the most likely pathways between one site and another, but they do not define how these pathways stand out over the entire communication routes. So what we want to define is basically the direct routes that stand out over the noise. And we base these on edge betweenness. So first of all, we calculate the number of shortest pathways that the bases we are interested in establish with all the amino acids and other nucleobases. And we call this noise. Then we compute the pathways from the sites of interest to the site of interest, and we define this signal. And then we calculate the signal to noise ratio as the distribution of the signal over the distribution of the noise. And this is the result. This is how it looks like. So imagine you have your noise overlapping with your signal. You will have low SNR. You will have a high SNR when your signal stand out over the noise. And this is, for example, what happens after target RNA cleavage. So before target RNA cleavage, when the system binds only to the guide RNA, the signal overlaps with the noise. But when you have the target RNA binding, the signal sourcing from this specific region that is called the switch stands out over the noise. And that's how we identified the region where to implement mutations. So once we knew that this region was very important for transferring the signal to the catalytic core, then we started analyzing what was going on around that region. And we identified that these mutations that increased the interaction with this critical adenine after target RNA binding. And then we worked on it, and we kept computing the signal to noise ratio in molecular simulations with these mutations, and Mitch performed the experiments showing that basically the cleavage product into these variants remarkably reduces and basically follows the trend that was observed in the signal to noise ratio, showing that these mutations have really something to do with the communications between the site of target RNA binding and the site of cleavage. So that's how signal to noise was designed.
Thank you. That was thorough. Thank you.
So Giulia, usually we end on time, but we are running a little over, and I do want to have a few more questions. I have a particular question. So we're very interested in how to use, how to generate models and then how to validate them against data. And one kind of validation or one kind of method you were talking about was NMR CPMG experiments, which are measurements of exchange broadening. And you pointed out that you have a network and you have different clusters or nodes, and then the links between the clusters you may get out of the modeling, but now you're going to sort of parameterize them based on the relaxation data. So my question is, could you use the relaxation data to validate the models? That is, can the models predict the exchange broadening effects? And now you ask, which of my various methods of simulating that match the data the best? And then related to that, and I'm trying putting two questions together. Can you use evolutionary covariance information as part of this analysis? And I realize that's a little different question, but so can you address those 2 points?
Yes, just one moment, please.
Like I'm imagining I have several different simulations from different groups, and I have data. So one is I can use the data to weight the link, the edges, but I want to use the data to validate the models. Is that possible in your mind?
So with CPMG relaxation dispersion with George Lisi, we had amazing results when we started the collaboration. We started in 2020. So what we observed, CPMG relaxation dispersion, so I'm not an NMR expert, but I like it very much. So I'm trying to study and understand well what, with CPMG relaxation dispersion, you can get an idea of the slow residues. So the residues that hold the slow dynamics, and according to the previous studies that were done on allostery, there is a review by Kern, the transfer of information is likely to happen through slower motions. So it is likely that these residues that hold the slow dynamics are the ones that transfer the information from one side to the other. And when we compared the residues that George observed to be slow in the dynamics of HNH, these residues overlapped for the 70% with the shortest pathways that we computed from graph theory analysis. So through graph theory, we computed the shortest pathways from one side to the other, and we had this very good overlap with CPMG relaxation dispersion, showing that the transfer of information happens through slow motions. But I think that certainly, I think that you can be very creative with these measures. And also what you said is something to be applied, to be tried. Maybe to use this data in a different way when analyzing the network. And this data can be all integrated in a different way. The fact of utilizing them in this way, I would say, is just one of the possible combinations that you can do between computations and experiment to combine the methodologies. I think that there's a lot of overlap that can be explored.
But let me ask the other question again. So if you have flipping between two states, some states have some contacts that are not present in the other states, and evolution may preserve both of those sets of contacts. So there may be evolutionary covariance that violates one state, but they explain the other state. Do you see any of that going on in your multiple conformational states?
I have not I have not investigated this, but this is a very interesting point. We are actually investigating now with NMR and MD a system that was obtained through directed evolution. And basically mutations appear along directed evolution in the domain, and this is something definitely to look at, I think.
Well, I ran the meeting over a bit, but I'll let, if someone else wants to ask Giulia a question or comment, it's open.
Hello. Can you hear me okay?
Yep, Alexei.
Yeah. Hi. I wanted to know in your communication analysis, whether it was necessary to include distance information between the pairs of residues that you're looking at, alongside the cross correlation.
Yes, because when you build a network, you give some cutoff. You give some distance cutoff and frequency cutoff. So the cutoff we are giving were tested. I tested them when I was a postdoc, and since they were converging, I never changed this. We never changed this cutoff anymore. So we are giving this cutoff, and the details are in supporting information of all the papers.
Would it be possible to ask a follow-up question? If there's time. Yeah, just that. So you mentioned that there's a cutoff and that this is a standard cutoff. I'll take a look at this at some point. But I've also read that in some other analysis of communication sort of propagation, that the communication sort of strength has like a 1/r³ relationship. And this is interesting to me because well, I personally it's nice to not have to use cutoffs if possible, to try and capture the most information. And I just want to know what your thoughts were on that.
I think that this is a very good point, but the reason why we need to use this cutoff is because otherwise we will get a lot of noise and we are not able to identify what are the relevant facts happening. It's like when you are having, for example, when you calculate correlation, when you do correlation analysis, you'll get these maps that are very noisy and sometimes difficult to understand and to visualize. Even to visualize, when you visualize a correlation map, you can see multiple things and you don't get the information you want. So the reason why you need to apply a cutoff is to get the relevant correlations, the one that are higher than 0.6, 0.7, and you need to do multiple tests based on your system to figure out what is relevant or not.
Okay.
Giulia, is a very,
much.
very relevant talk, and it's really beautiful work. It's, I think, really groundbreaking. You guys have done a great job bringing together experiments and simulation. I know there are people in my group who wanted to hear this, but they're on their vacation, and I think we had a smaller than normal group because of it's being July. So this will be available on YouTube, and we'll give you the YouTube first so you can edit it and make sure you're happy with it. So grazie mille. Thank you very much. Thank you for
Thank you. Thank you. Continue. It's wonderful work.
holiday. Thank you, Emily.
Yeah,
Thank you.
thank you.